What I want to do is talk to you guys as a community about what we need to know about clinical trials, how they get set up, and what we can start doing right now to help, and what we need to be doing in the future. And this is really a parent view. It's not an industry view. And I do have to say that I have a disclosure because I have helped Ovid in terms of setting up the STARS trial, and I serve on that steering committee. However, it's very, very important for you to understand that I do not speak for Ovid in any way, shape, or form. So anything I am telling you right now is my experience and what I think you should know. Probably most important is I am mom to Sophie, and on Facebook, this is what my profile picture looks like, so you don't know who I am, but you know who Sophie is. So that's just so you can maybe put me in context here. All right, so we're in a new frontier. When Sophie was diagnosed almost 10 years ago now, the research was just starting to take off and blossom, and it was amazing. And as a researcher myself, I was really taken by how much promise there was in the AS research field and how fast things were starting to go. And what's been amazing is now, 10 years down the road, we're at a very different frontier. And this frontier is that we've taken all that lovely basic research and we're starting to move into thinking about interventions and therapeutics, like you just heard from Ben and Stormy. And when we start doing this, when we start trying to figure out how do we take something and get it in, for at least in this country, FDA approved so that we can use it, there are lots of different steps to go through. And I'll just, as a disclosure right now, say I'm going to mainly talk, when I talk about regulatory things, about the FDA, because that's what I know. But in every country that you're from, there is a regulatory agency that helps oversee this, and they have their own sets of criteria, but they're not that different from this. So where we've been the last 10 years has been in the first part of this diagram, in the discovery science. So looking at things in culture, looking in basic research, looking at animal models, trying to get us to where we need to go. And someone asked the question, why is industry so interested in what we're doing? Like Ben said, this is a very tractable system, and it really looks like there can be a home run here. So we're very fortunate in that. And 10 years ago, one of the things we would say when we would sit around and talk as parent groups, we would say, like, all we want is industry to give a really good look to Angelman syndrome, because if they look really hard, they'll stay. And we've seen that. And what we're going to do in the second part of this is I'm going to have a lot of the industry reps come up, as well as some researchers, and they're going to give you an update on what they're doing in the Angelman syndrome space, and also take your questions. So where we're moving to now are clinical trials. And they have very different phases. In the first phase, it's very small, and what they're just doing is seeing what the drug does to someone who takes it. How fast do you metabolize it? Does it go to your liver? How much stays in your blood for how long? Do men metabolize differently than women? We know that's true. Do children metabolize a drug differently than adults? We know that's true. They have to do all that to figure out dosing. How much are they going to give you in a clinical trial? Once they have that, they can do a phase 1 study, so they can expand to more people and make sure that their dosing is good and that their safety profile is still safe. After that, you move on to phase 2, and in phase 2, you're still looking at safety, always looking at safety. But in phase 2, we're starting to try to figure out whether or not that drug actually has some benefit to the population that's being tested. And then after that, you move to phase 3 trials, where you have lots more people. You can already see a problem for our community. Lots of people who take the drug to look at how efficacious it is and how safe it is. So one of the big things that you need if you're going to test a drug and see if it works or not is you need to be able to measure what was the before and what's the after. And so you're going to hear a lot about this. You heard a little bit about it a few minutes ago. You're going to hear a lot going forward about something called outcome measures. And I know the first time someone said to me, we need outcome measures, I thought, what in the world is that? But it is exactly what it sounds like. These are ways to measure the outcome of your trial. And they have to be tests, and they have to be accurately assessing the function of an individual before the trial begins. So for our kids with Angelman syndrome, we need to be able to see, where is that kid? Take a snapshot in time. Where is that kid right now, or adult or teenager? I apologize to the adults and teenagers if I'm leaving you out. Where are you at that step in time? And then can we look at you after the treatment and see, have you made any progress? So those are outcome measures. And you guys know what these are because you've probably been through them in clinics and things like that. These are the tests. Can you put pegs in holes? Can you stack blocks? How many words can you say? How do you as a patient feel? Are you stressed? How do you as a parent, are you getting any sleep? These are all outcome measures. They can be used during and after treatment to assess whether or not the treatment works. And this is one of the big things we have to do in our community, is we have to push and develop really strong objective outcome measures. You're going to hear a little bit from Terry Jo Bichelle in a minute about ABOM. This stands for Angelman Syndrome Biomarkers and Outcome Measures Alliance. This is a new initiative, and she can tell you a little bit about it, but the whole point of this initiative is to make sure we have those outcome measures so that our trials can be successful. All right, so this is the audience participation part. So you guys didn't know this was going to happen, but I need all of you to help me out here, because what we're going to do is we're going to design a clinical trial. And the purpose of this is so that you guys can sort of see what goes into this, like what kind of things do you have to think about, and I think this will help you understand what these trials are like when we actually start participating in them. So I'm going to give you the hypothesis, because I've been studying this for a very long time, and I have the thought that parents of children or adults with Angelman syndrome drink more coffee than people who don't have kids with Angelman syndrome, right? This is my thought process. And I feel like I have really good basis for this in basic research. I think it's because our kids make us tired. And, you know, that's just how it is. And so that's going to be my test question. Of course, it's a ridiculous test question, but it works for this particular analogy that we're going to do. All right, so this is where you guys come in. How do I test this? How am I going to figure out how much coffee you drink if you have a kid with Angelman syndrome or not? Really simplistic. Somebody shout something out. I could do a survey, right? I could send a survey out and say, how many cups of coffee do you drink? How else could I do it? Do what? Watches? Watch you, yes, absolutely. Yeah, hi Billy. Billy likes that one, yeah. So yeah, we'll put cameras in your house and watch how much coffee you drink. We'll take notes, we'll have videos of you drinking your coffee. Yeah. Say it again? Do a blood test. How much caffeine is in your system? All right? These are all things we can do, so we got to think about which one of these is the easiest, the best, the most effective. All right, so my study subjects are pretty easy, right? I'm going to take parents who have kids or adults with Angelman syndrome. What am I going to compare you to?
Parents who don't have Angelman syndrome.
Parents who don't have kids with Angelman syndrome. Okay. How many, what do you think, Billy? Is that good? Yeah, okay. Billy says it's good, we're good. How many of you in this room do not have a kid with Angelman syndrome? Okay, you're my control group. You do not have, how many of you don't have an adult? You don't have any family member with Angelman syndrome? Okay, good. You're my control group. All right. So now we have a control group and we have our test group. Now here's the thing. We want these groups to be as similar as we can possibly make them, right? How do we do that? How do we make the control group and the comparison group as similar as they can possibly be? And the reason for this is, think about it. If I compare David back here, who has a very young child with Angelman syndrome, to say Linda, who has Billy, who is a very handsome young adult, maybe they're going to drink different amounts of coffee because of this, how old their kids are. So how do I compare that to our control? I also have to make sure that all of you who raised your hand and said you don't have a kid with Angelman syndrome, do you have kids at all? How many of you don't even have kids? Yeah, I don't want you in my control group. That's not going to be a good comparison. You're not going to drink coffee. Okay, how many of you don't drink coffee? I don't drink coffee. I don't want you in my groups at all because you can't answer my question. And the problem is, if I put you in my groups and you fill out your surveys and things like that and it goes into the numbers, I'm not going to see differences. They're going to start to get washed out because there are people in my study that don't fit the criteria of the study that we're having. Does that make sense? You see some nods? Okay. This is how complicated it gets, right? I want everybody to be as similar as possible. Like, in the perfect world, we would clone everybody, and one of you would get the drug and the clone wouldn't, and we would see what the difference was. We can't do that yet. So we need to match things as closely as we can in those two groups. That's where you get the inclusion and the exclusion criteria for clinical trials. They're not there to make anybody feel bad. They're not there to prevent you from having an opportunity. They're there to make sure that the trial is successful. So this is a very basic trial design. There are tons of trial designs, and I started to put them in here and thought, we really don't care. But the easiest trial designs are you take your population and you split them into a treatment group and a non-treatment group, and then you give them the outcome measures and you see what happens between those two groups. And you try to match them as much as you can, and you want to see what happens. So one of the things we have to do with our control group, besides matching it, making sure our kids are the same ages as much as we can, they have the same mutations as much as we can, we also want to give a placebo. And whenever we say placebo, people go, oh, I don't want to be in the placebo group. Nobody wants to be in the placebo group. Do we even need a placebo? So I want to talk about this really carefully because this is so important. Why do we have to have a placebo? And I'm going to subtitle this, I know my kid, I know myself, I'm going to know if something's different. Of course you are. Of course you know your kids. Of course you know yourself. That's completely understood. The thing that's really bizarre about what we call the placebo effect is that you have absolutely no control over your brain in this particular situation. None. You think you do. You absolutely don't. This is what it is. Your brain gets a new experience, in this case the drug, and this really freaking weird thing happens where your brain tells your body what's going on and you respond to it. It's really amazing. So here's an example of this. There was a clinical trial that was done on acupuncture, and they found out that acupuncture really did work. People who did acupuncture got relief from pain. What was interesting about it is it didn't matter where you stuck those needles, it worked, right? It didn't matter if you put them on the points you're supposed to put them on. So our brains are amazing, amazing, amazing things. And the placebo effect is such a problem in clinical trials, and in fact, the head of Merck came to my university to talk about his neurological treatment program and said that this is how they feel most of the time. They're like, why aren't we just giving people placebos? They work better than anything we try. And again, this is not a judgment. You have no control over this. This is just how our brains work. And I'm sure evolutionarily, there is some reason why our brains do this for us. I mean, it's good to be better. It's good to get over being sick. And you may think, how can you have a placebo-controlled trial? It's my kid that's getting treated, not me. Well, you're going to pay more attention to your kid if they're getting a treatment. You might try things you wouldn't normally try because you know your kid's getting a treatment. This is not anything that you're doing on purpose or not on purpose. It just has to be done. Does that make sense in terms of the placebo? All right, so here's the flip side of the placebo. It's called the nocebo. I had not heard of this. The nocebo. The nocebo is when something harmless causes you harm because you think it's harmful. It's the flip side of the placebo. And Dr. Anton gave me permission to give you this anecdotal experience from the levodopa trial that he conducted, and my understanding is that publication is going to come out soon. The most adverse side effects in that trial were reported by people who were on placebo, and in all cases, those people said they were absolutely sure their kids were taking dopamine because of how adverse the side effects were. They were on placebo. So it's the same thing. Your brain does this. And I'm going to give you an example of why your brain can do this. I'm going to hope this is going to work. So here's the situation. You're camping, you're out in the woods, you're in a lovely cabin, it's getting really, really dark, and you look out the window and you see this large, dark shape lumbering slowly across your backyard. What do you think that thing is? I hear bear. How many people think it's a bear? How many people thought it's probably a bear? How many people thought it was a polar bear? Billy thought it was a polar bear. Okay, none of you thought it was a polar bear, but a lot of you thought it was a bear. Nobody thought it was a Tyrannosaurus Rex, right? And that's because our brains are pattern generators. They take information from disparate sources and they put it together and they come to conclusions. The fact that you're in this room with all of this stimulation, me talking, slides, lights are on, people are talking around you, and you're processing all of that, that's amazing. And you can take information like this, and you can put it together and come to a conclusion very quickly. That's, again, it's something that lets us survive, but it gets in the way when we try to do clinical trials. So what do? We've got these amazing brains that are getting in our way. What are we supposed to do? And I'm taking this from someone at the recent scientific conference that we just had because I thought this was great. This is what we do. In God we trust, everyone else bring data. We have to measure it, and it has to be objective. It can't be what we think as parents, because it's not reliable. It has to be something that's reliable. So what are some of the difficulties in this? You heard a little bit of this from Ben and Stormy. I love this cartoon. I like this cartoon in general, but I like this particular one. We're a rare disease. We have small numbers of people. So When we were trying to approve something like Wellbutrin for depression, for example, the effect of Wellbutrin on people who were depressed was very, very small. It was significant, and it really did help people, but it was a small effect, and the only way you could see that was to have thousands upon thousands of people in clinical trials so that effect became apparent and became statistically significant. I am completely and utterly deficient in math, so I am not explaining statistics to you in any way, shape, or form. But the bigger your group size is, the more likely you are to see something subtle. We have a small group size, that's just how it is. And so when we design clinical trials, we've got to think about measures that will let us see subtle effects. Because if company X has a drug that lets my daughter learn something twice as fast, I want that drug, right? If she could learn things twice as fast, oh my God, the things that could happen in my house. So I want that drug, but that's going to be subtle. How are we going to see that? The other thing we have to think about, and Ben mentioned this, is the fact that our kids are pleiotropic. You might ever hear people say our kids are heterogeneous. What it basically means is our kids are all different. I'm sure every one of you out in the world could see a kid walk by with Angelman syndrome and be like, that kid has Angelman syndrome. There's something distinctive. We can tell when a kid has Angelman syndrome. But they are all unbelievably different. And you don't have to read this list. This is the clinical criteria for Angelman syndrome that you're all aware of. But our kids are very, very different too. And I have permission for this example. Erin Sheldon's coming to do the communication groups. Her daughter Maggie and my daughter Sophie were born about the same time. They're about the same age. We know their break points. They have the exact same deletion with the exact same break points. They're missing the exact same amount of DNA. Maggie met all of her motor milestones on time because she has hypertonia. She walked on time. They thought she had Prader-Willi because she was meeting her milestones. Sophie is 11 and she still can't walk. They both have seizures, but Maggie has partial complex seizures where she throws up a lot, and Sophie only has atypical absence and drops. Exact same loss of DNA. I could go on and on and on with this, but you guys know this yourselves. Our kids are all different. So how do we measure something that's improving in our kids if they're all so different? So what do we need to have happen? We have to have objective, measurable outcomes. They have to be robust. They have to tell us what's going on in a very small when we have small numbers of participants, at baseline and after treatment. We also need to determine why it's going to improve our quality of lives and our kids' quality of lives most importantly. Ben mentioned drooling, which is a great one. If somebody comes up with a drug that helps my kid chew chewing gum, I really don't care, right? Fantastic, thank you, it was a great improvement. I don't want chewing gum in my house. It's a ridiculous example, but there are some things that maybe we can improve that we don't care about and that any kind of adverse effect isn't worth the risk. There are things we do care about, and the companies want to know this, and they're doing surveys and things like that to get more of an understanding of us. Now you may think this is obvious, but only recently are the researchers cluing into something that we as a community have known for a long time, and that is some of our kids do not eat. They look at food like it is poison. And then on the other hand, we have kids that are very, very like Prader-Willi. They will not refuse food if they see it. A lot of researchers didn't know that, but the parents all knew it because we talked about it all the time on Facebook. They now know it, and there are studies that are going on. So they need to know from us what's important. We have to make sure these trials are carefully designed so that everybody's time is well spent, especially those of the individuals with Angelman syndrome that have to participate. This is critical, critical, critical. We have to have strong cooperation by all of the stakeholders. That is the patient with AS first, caregivers, all of the support and research organizations around the world, the researchers and industry. We have to cooperate. We can't pretend like there are divisions. We can't, any of these little dramas that we like to go around with, we gotta get rid of them because we need to all be working on that goal together. We're just too small not to do that. All right, what can you do as a community? Participate. Ben and I might differ here. I would say don't cherry pick your studies. There's no telling what's coming or not coming, right? We don't know. Companies run out of money sometimes and something really promising stops. We have to participate. And I love this cartoon of this guy saying, oh, they're doing a great new test for this cancer that I have. And she says, are you gonna participate? And he says, no, why would I do that? And she's like, yeah, me either. I hope they get a treatment soon. There will be no treatments if we do not participate in the trials. That's just the bottom line. How do you find trials? Clinicaltrials.gov is one very good place to do this. You can't see it, but I've put Angelman syndrome in there, and it's popped up all the things where you can be involved if you have Angelman syndrome, and the trials from Ovid are there. Sign up for the newsletters from your parent organizations, sign up from the ABOM, sign up from anything you can that will give you these newsletters and read them when you get them. Get involved in the Facebook communities. We have a research community, we have connections, we have all kinds of different groups, and all that information will be there. You should get informed about trials in general. I'm giving you my take on this, but the FDA has a website that's all set up for patients. And you can go and read about what they are, how they're conducted, what your rights are as a patient or a treatment participant. You need to be informed. And when you get ready to do a trial, they'll give you a big long document that tells you about the pluses and minuses and all the things. Read it, be informed. This is a bizarre one, but bear with me on this. Don't take things too personally, but educate. This is what I mean by this. You're all familiar with those tests that we take when we go to the clinics and stuff like that. And I'll be honest with you, I hate them. Hate them, hate them, hate them. I do not like going somewhere and looking and thinking about all the things that my kid can't do. We have to realize that we can't take these things personally. All they are numbers. They're numbers to say your kid is at 12, and after the treatment they're at 15, and that's an improvement. That's all that matters. And I'm really pushing hard to get clinicians to stop putting age equivalents on these things. Our kids are not babies in big bodies. And when you tell somebody their kid is functioning like a 24-month-old, that's ridiculous. They may be at a 12 on their fine motor skills, they're not 24 months old. So we can't take it personally because we have to have those measures, we have to have those numbers. So I'm going to give you an example of this. This is the terminology that I like and I push. I'm really annoying too. You can ask the researcher, they get really annoyed with me. I prefer that we say that our kids are either strongly affected by AS or they are less affected by AS. I really hate people telling me that my kid is low functioning or high functioning. What does that even mean? It's a very small difference, right? We're just changing the words, but it means something different to say I'm strongly affected by this, or I'm less affected by this, or I am low functioning. It's horrible. All right, and I have permission for this. I'm going to show you a really low functioning, supposedly, kid with Angelman syndrome. I prefer strongly affected, but here's some of the criteria and those tests. Seizures started at 7 months of age. This kid didn't sit up with support until they were 3 to 4 years old, so they met that milestone late, even for kids with Angelman syndrome. Can't walk, has tried up to 5 seizure medications and is currently on 3 to keep their seizures at bay, and has the lowest possible score you can get on one of these tests, whether it's fine motor, gross motor, communication, whatever it is. This is one of the lowest scoring kids possible on this test. All right, now brace yourselves because I'm going to show you this kid. Here's your incredibly severely affected kid with AS. This is my daughter, Sophie. She's horribly affected, but she's hell on wheels on that bike. And what you can see here when you watch this video is right here, see how she slows down? She's scoping out the toddler that's walking over next to the playground because she likes to run over kids with her bike because they make really great noises. And so if you watch her as she's biking, you'll see that as she comes around and sees that kid, she gets a big smile on her face. So, like right here, she sees that kid, she's like, ha 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 ha, I see the kid. And I'm with the camera saying, Sophie, do not go over there. And at this point, she completely ignores me and goes after the kid, and I have to drop the camera and go get her. Strongly affected kid on all of the tests that there are. Come on, right? So that's executive planning, that's functioning, that is deviousness. That is not a 2-year-old in a big body. So we can't take these things personally, we gotta get those numbers, so just take the tests, throw the age equivalent thing out of the park, and just say, these are numbers, these are things to improve on, I can't wait for the drug that's gonna make these things improve. We gotta think community-wide about this, we cannot get in our own way. And what that means is when there's a chance to participate, we need to participate. And when I say don't get in our own way, I'm gonna tell you another story from Liz Berry-Kravis about the Fragile X community, and she gave me permission to do this. They had a small clinical trial, phase 2 trial with a drug that was working. They thought for sure it was working. They were getting some positive benefits. The parents that were in that first trial were thrilled, kept their kids on the drug, were really excited. Everybody was excited. So they started the big trial, where they were going to have lots of people involved in the trial. And the Fragile X community all went on Facebook and figured out how to take the intake survey for the trial so that they could all get into the trial, even if they didn't meet the inclusion criteria. Right? So if you took the survey a certain way, you got a score that made you either in or out. They figured out how to take the survey. They all took it. They all got into the thing. Now, like we were talking about planning that trial before, now we have people that shouldn't be in that trial. We have the non-coffee drinkers in our coffee trial. And when they did the trial, they couldn't see the effect in the bigger group because it was the wrong group. The problem with this is the company took the drug away from them, so all the kids that were taking it and had benefit had to quit. And they never got that drug, and they still don't have it. We can't do this to ourselves. So if you meet the inclusion criteria, fantastic, join in. If you don't take it personally, you can't be in this trial. But if we do the trials well, and we participate the way that we're supposed to do it, everybody will get the drug once we show that it's effective. But we're not going to get it if we can't do that. Ben mentioned study fatigue. We're going to get tired. We're small. We're going to get tired of the surveys, tired of the trials, tired of the things. We'll push. We'll try to do it. We'll try to spread it out so that we don't all do the same ones and things like that. Somebody asked, what happens if you do one? Can you not do the next? It depends. They're not going to be able to exclude all of us. How many of you were in the betaine folate trial with your kid when that was going on? Not too many. Okay, but if they excluded all of us, there would be a group between the ages of 7 and 15 that couldn't be in any clinical trial going forward. They will ask you sometimes to come off of something you're taking, to come off of maybe CBD or curcumin or whatever the thing is. Again, that's to make those groups as similar as possible. Just do it. Take the 3 months off whatever supplement you're using if you can, and just do it. Document. You probably have stacks of papers about your kids. I do. Keep them, because they're going to ask you questions like, when did your kid first pick up a Cheerio? I don't know. But you probably do know, because when you filled out your early intervention papers way back when, it was very clear in your mind when that was. You have those papers. Keep them. Get that information to the researchers. Visit the AS clinics. They are worldwide. There's a phenomenal one in Erasmus. There's a great one in Israel. We have lots of them in the US, lots of clinics. Get that information to them. Win Hontan is going to come up and talk a little bit about what's going on, but we're going to have a new natural history study. It's going to be new and improved. That's a way to get it to them. We need a natural history study because we need to know what the progression is of AS in every single subtype. What does it look like if you're UPD from birth through 25? And we need to know which of those tests are the robust ones, and we can do that by having you take them when you're in the natural history study, and then we have all that data for the industry to look at and say, wow, that looks like a really good test, I'm going to use that one. And one of the pluses to this is if they have lots of great data from natural history studies, they can often reduce the number of people they need on placebo because they have lots of baseline data already. So that's a win-win for us. What can you do this week? On Thursday, we're going to have a focus group for the new natural history study at 5:45. We want everybody to come that wants to come. This is going to be in Camelback Room D, and what we're going to do is we're going to talk about the history study and how it's going to be set up and what we're going to measure and all these things. Are there people here that would be interested in participating in that and making sure we do that history study in a way that's good for our kids and good for us? Fantastic. Please come on Thursday, tomorrow. If you've got a minute right now, pull out your phone and go to www.angelmanregistry.info. Click register now, enter the very basic of details, and then confirm your email when it comes through that you're registered. That's all you have to do. Just register. This is the global Angelman Registry, and we're trying to get everybody in the world to use it, put all the information into it so that we can query something. And what I mean by that is a researcher can come on and say, how many kids with Angelman syndrome are taking melatonin? And this registry will de-identify your data. It won't say Sophie's taking it. It will say an 11-year-old who's deletion positive is taking melatonin. And it will tell them all that information like that. You have no idea how amazing that would be in terms of setting up trials and making sure people are looking at us. All right, so thank you for listening to me ramble on and on. What we're going to do now is have the reps come up and give you a little update about what they're doing in the Angelman syndrome space, and we're going to open it up to all of your questions. And so hopefully everybody's here. I see Megan. Is Jody here? All right, so while they're all coming up so they can be on this panel and you guys can listen to what's going on, I just want to mention that we're used to the academic researchers in Angelman syndrome saying whatever they want, whenever they want, as loud as they want on Facebook, whatever. The rules that apply to academia are very loose. The rules that apply to industry are very strict. The FDA pays a lot of attention to what industry does. And so there may be things that you want to know that they just can't answer, they're not allowed. So it's not, we don't want to participate, it is, we can't answer these questions. And so I assured them all on this panel that if they can't answer, they can say so and we will understand. With that, Ovid can't be on the panel because they're running a clinical trial, and so therefore they're not allowed to do this. They are allowed to have a booth, and they have it, and they said, please come by and talk to them. 2 questions I can answer about the Ovid trial. Are they still recruiting? Yes. When will you know the results of that trial? When they get enough people in the trial to be able to tell us something statistically significant. So go talk to Ovid at the booths. All right, so we'll start over here with Jody, and we'll let her update you on what's going on with them.
Great. Thank you, Becky. Hi, my name is Jody Cook. Oh, do I need to
Push it.
Okay. Oh, much better. I'm Jody Cook. I'm the Chief Operating Officer of Agilis Biotherapeutics. Thank you very much for inviting me here today. Agilis has been working in Angelman syndrome for a little over 2 years now, so I feel like the old timer in the group. We are working in gene therapy for the central nervous system. So in addition to Angelman, we have 2 programs that are further along. One is an AADC deficiency where we're in a phase 2B, and we have 18 children with anywhere from 1 to 5 years worth of data. We're in Friedreich's ataxia where we're in IND enabling studies, so we're really in monkeys. And our Angelman program is about to select a lead for gene therapy. So what does that mean to you? We have been studying over the last couple years variations on the gene, the promoter, all these kinds of things that you put around the gene, and what route of administration, how do you get it to the right cells? So we're pulling that data together, and our goal is that at the end of this summer that we'll select a lead. And so what that means is you freeze the design of the therapeutic, and then you take about 18 to 24 months and you go through very structured studies that are required by the FDA to get into human clinical trial. So anybody that gets into a human clinical trial, they've done a number of studies to prove that to the best of our ability that the therapeutic is safe and has a likelihood of being successful. So to what Becky had said, I also echo the what can you do today? Participate in the outcome measure studies, the natural history studies. 2 years ago when I joined in with this group, I think I said to everybody I met, the most important thing you can do today to help anyone with a potential therapeutic is share that data. If you live far away from a clinical trial site, that's fine. Get on the registry, get online, so you can participate in various ways. And so Agilis listens to the parents, listens to families. We listen to what is important to you, and our decision making in which therapy to take forward is it going to target a potential meaningful outcome? So I'm delighted to be here. Thank you.
Hello, my name is Megan Miller. I'm a scientist with a background in neurobiology and a project leader working in the discovery unit at Roche in the rare disease group. And today I can't talk about what we're doing specifically. We do have a preclinical program ongoing. I can say that we are aiming for transformative therapies in all of our rare disease programs. So this is the underlying goal that we are trying to achieve in all the diseases we work in. What I do want to emphasize is, again, that similar to what you've been hearing over the past hour or so, is that it is very important, even at early stages, to start to understand what the relevant clinical outcome measures are, what we can expect to see change in clinical trials. And to this end, Roche has committed some money, some funding to the community to instigate the initial stages of what we're calling a disease conceptual model. And what this is it will be a very comprehensive document that will inform us very robustly about the impact of Angelman syndrome on the lives of both the patients and the caregivers, in a way that we can use this information moving forward in identifying fit-for-purpose measures in our clinical trials, what we should be measuring, and that together with the natural history studies will really be what allows us to develop efficacious treatments.
Hello, my name is Mark Leitchman. I'm from Ionis Pharmaceuticals in clinical development, and we are also in preclinical development, so not quite ready to go to clinical studies, but we're also committed, just like all the others, to bring our drug to the clinical studies and hope to go forward. And yeah, I think most of the important things have been said by all the other participants here. I also think that it's important to understand, you know, we all wear different hats, but we all have to kind of work together to bring this forward. I think as a community, you're in a great position to be in. You have multiple companies having multiple compounds that address multiple needs, multiple ages. That's really where you want to be. And I think there's a very high chance that over the years, these drugs will come through and provide some benefit to them. And generally, I think, you know, my recommendation would be stay engaged. You know, speak up if there's something that you like, if there's something that you don't like, and you know, also whatever you do, it has to work for you. So you know, while we like you to participate in studies and so on, it has to work. If it doesn't work for you, that's not right either.
Hi. Hi. So I'm Win Han Tan from Boston Children's Hospital, and we have been running a few studies. We have been running the natural history study initially from 2006 to 2014, and hopefully we are hoping to restart that in a few months. So we really look forward to all of you participating. Unlike the previous natural history studies, we are going to have more study sites across the country, so you can go to any of the study sites that's close to you. The way we like to see ourselves in relation to the other pharmaceutical companies is that we are helping them provide data, the outcome measures that they can use to run the clinical trials that we so need in this community. And we also provide the infrastructure that allows them to launch their clinical trials efficiently. So I think Becky has done a phenomenal job just now telling you about the benefits of participating, and I would actively encourage you to do that. And if you can come to the focus group where we'll talk about some of the details of the new natural history study tomorrow evening, that would be wonderful.
Just to follow up, as the community, we need to make our voices heard. And so when you are asked if you would like to serve on a steering committee, what that means is having your parent voice in with maybe a company doing clinical trial design or the natural history study, making sure we're doing the right things and we're figuring out how to structure those visits well so that they get the best out of our kids.